Hi, my name is Meera Baladandapani. I'm a professor in the Department of Biostatistics and hold a courtesy appointment in DCMB as well. And it's my distinct pleasure to introduce today's speaker, Kahan Liang, who's the Bernard Family Distinguished Professor in Targeted Therapies and the Deputy Chair of in the Department of Bioinformatics and Computational Biology at the University of Texas MD Anderson Cancer Center. Dr. Liang is an accomplished scientist. He joined MD Anderson after completing his bachelor's and PhD in Princeton, and has been at Anderson, 10, 12 years now. And he's risen really vertically and has touched many areas of science, especially in terms of cancer genomics. And his main thrust of his work is on bioinformatics tool development, and has developed many, many tools that are used by over 100,000 users across the world, including the Cancer Proteome Atlas, TANDRIC, FASMIC, and some of the DrBioRight, which I believe he's going to be talking about today. And he has published over 140 papers, and which have been cited over 25,000 times. And I can claim to having co-authored on 2 of those, at least, not in terms of citations, and not so high in terms of citations. And that includes like the top-notch journals such as Cell, Cancer Cell, Nature Genetics, Nature Biotech, and Nature Med. He has taken on leadership roles in many consortiums, especially the Cancer Genome Atlas, the Pan-Cancer Atlas working groups, and as well as his list of awards is a long, winding list of awards, and that includes many awards within the University of Texas system, as well as being a fellow of the American Association for Advancement in Science, and as well as the AACR Team Science Award. So without further ado, I would hand the floor to Dr. Han. And we would have loved to have him in person, but I'm sure he's enjoying the 60-degree weather in Texas versus our 18-degree and over weather. So Han.
Yeah, thank you, Vera, for very nice introduction. And I really miss you. And then you are such great collaborator and friend. So hopefully one day we can have this in-person meeting. Today it's really my great pleasure to join this seminar series to give a talk on talk a little bit about the fundamental things in this big data. So artificial intelligence, big omics data, and the cancer system. So you can see my slide. I try to. So this is my disclaimer. So for my work with our company and software, and then this work I present, part of is in collaboration with NVIDIA. Okay. So I think we all now agree that we are in a very exciting time of big data. So in the last 10 years, what you see is with the next generation sequencing, the tremendous molecular data has been accumulated. So this from the genome, transcriptome, proteomics, and more recently you see single cell sequencing data. So I think we are in a very exciting time for discovery. On the other side, we all know this also have a lot of challenge how to interpret this data. So in the last 10 years, my group have focused on a big consortium data called the TCGA, or the Cancer Genome Atlas. As you probably all know, this is really provide a comprehensive molecular profiling for more than 11,000 patients from 33 cancer types. So for each cohort, usually more than 500 patients, you generate this multi-dimensional data set from mutation, copy number, gene expression, DNA methylation, even micron expression, and also protein expression and clinical data. So when you have this multi-dimensional data over the same patient cohort, this just provides a tremendous discovery power to understanding the molecular mechanism, and also try to identify some clinical biomarker and new therapeutic target, even design new therapeutic strategy. So in the last year, 2018, when we finished the TCG Pan-Cancer Atlas, we invite to write a paper in Cell. So in that Cell, we talk about the long-term impact of TCGA. So really, when you have hundreds of scientists working together over such big data set, what really, I think one key point I want to mention is through large consortium projects such as TCGA, essentially put bioinformatics and computational biology really in the driver's seat of the cancer research. So personally, when we think about bioinformatics or computational biology more like a supporting, we are get some, do some analysis suggested by our collaborator, but really with this huge data available, data generation, data resource is no longer the bottleneck in cancer research. It's really how to use fascinating bioinformatics tool, cutting edge algorithm to convert this big data into clinical and biological insights. So of course, when we talk about the big data, it's not only TCGA. It's really you have the international ICGC, right? You have 1,000 genome. And then we also have many big consortium like GTEx, which provide normal tissue gene expression data, ENCODE, which provide comprehensive regulatory elements in human genome. And then recently you see UK have this biobank where you have 500,000 genotype phenotype cohort. And also US NIH started this All of Us, the precision medicine aim like 1 million people how systematically collect the molecular data and then the health data. So this all kind of data fundamentally on how to use basic data to generate clinical actionable on very practical for health care. So when we talk about this data, of course, this big data present big challenge for bioinformatics, for computational biologists. So I think today, so really what we need is two things. Number one, we need a more effective bioinformatics tool to analyze those data. Second one, we probably need some fundamental new analytical concept to understand the complexity of the biology and the disease. So my group actually work on pretty much in that to address those questions. We work on the intersection between bioinformatics, computational biology, and systems biology. So over years, my group published quite a bit bioinformatics tool, try to help people analyze those large-scale cancer genome data. And then we also perform a lot of pan-cancer analysis, which means we focus on the same question. We ask the same question in different tumor contexts, try to, through this contrast and comparison, we try to understand the common principle and the cancer-specific feature. So those cover a broader range of topics from pseudogene, RNA editing, sex effect, to more recently mitochondria and the enhancer. And then based on that, a third component in my group, we also developed this high-throughput system biology approach through those combination of cell assay and then functional proteomics. We try to understand the driver events of mutation, gene fusion, and try to design new therapeutic drug combination. So really, that's a big concept, that picture of my research. But today, I will give you two topics. One is really focused on bioinformatics tool development. I hopefully introduce this DrBioRight, a conversational AI-driven analytic for omics. The second topic will be a more computational biology side. It's really a pan-cancer analysis about the enhancer. So we focus on enhancer RNA. I propose that this will be very exciting paradigm for biomarker discovery. So the first part, when we talk about the big data analysis, it's not only the data size is big. So when we talk about data now, it's not a megabyte, it's terabyte, even P. So when you talk about this data storage is a problem. And more importantly, there are so many diversity of data. You have different data type. For each data, you have different analysis. For each analysis, you have multiple algorithms. And then no matter how you try, there's always a learning curve, especially for those people with no computational background on the primarily work on experimental side, on clinical side. So there's a technical barrier. Finally, when even you spend the time, learn some tool, become the master, the whole field is just moving so fast. You always have new algorithm, new data sets. So even just keep tracking on the recent development in the field, it could be very challenging. So that's really urge us to think about how to fundamentally change our practice in data analysis. So the other big thing you probably all know, nowadays people have deep concern about reproducibility in those papers, especially for those papers published in top journals. You see a lot of papers published, then they get retracted, or some paper published, they found they cannot be reproduced. That will be problematic. So our department actually has this very good reputation, try to check reproducibility. So that's also another fundamental issue in data science. So ideally, we want to have this kind of ideal platform. Number one, really want to minimize the communication bar between user and the tool. You want to make this lower and possible. Basically, you want people have no background in computer training can still using tool to do some work. Number two, we want to that result transparent, reproducible. So which means we do something not only give you the result, but also have you detail to reproduce that. So essentially, we want to remove the black box. Third one, we want open development. We want to have the tool engage the whole community to contribute the tool. And more importantly, we want to that tool, it's not static. We can really, based on the user feedback and the user's data, to constantly make progress, to learn new ability, to make a better judgment for your analysis. Okay, so that's really, so my group in the last 3 years, 2 to 3 years, we really focus on those concepts. We guided by those principles, we developed a tool, this tool called DrBioRight. So this is the first natural language oriented analytic platform for us to analyze omics data. So I can show you the video. So here, this tool actually has extremely simple interface. It's really just like a dialogue, just like what you use in social media, like Facebook, WeChat, you have this WhatsApp. So you only have this one input area where people just can type a sentence directly in this area. Say, how many you want to ask a question, say, how many cancer types do you have in TCGA? That's even simple. And then the analytic DrBioRight essentially understand this natural language. They ask you to confirm, do you want to have summary about TCGA cancer data? Once you confirm this yes, then DrBioRight will go to the literature, go to the data cohort, get the analysis, generate this bar plot where you not only know the cancer type, but also know the sample size for each of this cohort. And of course, you see you can also download this figure for your own analysis on presentation. For example, you can ask the question, could you do a correlation between, say, between TP53 versus P10 gene versus BMR2 gene in TCGA. So in this case, you're interested in breast cancer. So in this sentence, actually, you did not specify which molecular data type you are looking for. DrBioRight will ask you which data type you are looking for, and you say, oh, you want to looking for the correlation between gene expression. So it's a gene expression. Then DrBioRight got this idea and ask you, do you want no test association between the two genes? Now immediately you see they can generate this scatter plot where they show this 2 gene P10 really, and then BRM2R show very positive correlation in breast cancer, and then you can download that figure. And also see DrBioRight also asks you to rate this job. So whether you're satisfied with job. If you're satisfied with job, you can just say give yes. And then if you're not satisfied, you just give a bad. This feedback essentially help us improve our underlying model, how to interpret your sentence. So here, basically, you have another task. Say, how about to do a differential analysis between gender? So you're interested in female patient and male patient in kidney cancer. You just type this sentence, and then Dr. I try to understand and say, do you want to do association between gender and GFG? See, you get this result in this kidney cancer. This gender female have much higher expression of DNF gene. And then don't forget to give us feedback, you sum up. That's good tuning. So you may also just want to do a quick view about the mutation. You want to identify some, say, survival biomarker. Could you just say, you do a overall survival analysis TP53 mutation, gene expression in breast cancer. So when you just type this sentence, you did not specify which data source. But in our platform, we already have TCGA several sources. You may choose TCGA. Now, Dr. Wright know you ask, and then the data set will test. They will directly generate this kind of do the analysis for you. So immediately you can get a figure, see Kaplan-Meier curve, where show you in TCGA breast cancer TP53 high expression show much worse survival. So this kind of analysis, of course, you say if you give a capable bifurcation, they also can do this. But what I mean is through this kind of natural language driven AI module, you could do this, get result just in a few seconds. So you don't really need to, for this kind of routine bioinformatics task, you don't really need to contact your bioinformatics collaboration, where usually you have to start email, begin to schedule meeting for the background. So one last example, you want to say, hey, could we do a mutation view about TP53 mutation in TCGA kidney cancer? So immediately you get this lolly plot where you show all the mutation in TCGA, how they distributed across TP53 gene. So again, you can give a feedback. So this just show you a little bit idea how this tool, when you're using natural language, you can directly initiate some bioinformatics analysis, just like you talk to your bioinformatics collaborator to do the analysis. So actually, DrBioRight can do that more than that. For example, it's not only can do some higher level analysis based on standardized data, you also can, one challenge, it's really, we can do some low level analysis. For example, if you started from some next generation sequence raw data, you want to do, say, you ask Dr. Wright, could you do a mutation calling? And then Dr. Wright will ask, okay, you want to do a single mutation only on your have matched tumor normal pairs, and you say matched pairs. And then Dr. Wright will ask you, where could you tell me the data set? For example, you are interested in a data set where it's publicly available in a NCBI SRA archive. You just provide the ID there. And then Dr. Wright will ask you a few questions about what tissue you type tissue, and then could you give a name for this product? Then you begin to do this. They ask you, you want to do the mutation calling based on that sample, based on that whole exome data, then you confirm. So in this case, this kind of data processing may take some long time. Then you can take a break. So you can enjoy a cup of coffee. Dr. Wright essentially using some cloud resource to this mutation calling. When the time ready, they will send you email say, hey, your data available. Then you can go ahead to click the button to see. They basically show this data report. In this data report, you see on the left side, they give you the index of what we report. And then you can go through those general statistics, say how many samples. So in this case, we have 5 samples, and then you're looking for some feature, how many, what's the base, how many reads are there. And then you can also looking for some sequence quality, say FASTQ, QC sequence quality, if this good or bad. And then you can also looking for sequence score distribution. Of course, for each figure you're looking for, you can always download that, save that for own future use. And then you can also looking for quality score distribution. See, generally those rates show very good quality. And then base content, you have ATGC, see nothing special. You have GC content you raise, so you see that pretty much fit you you can try different interaction, and you can see the base and content. So there's no much not result base, and then you can see the duplication level, and then over represent the sequence. So these are all like normal some procedure. When you get data, before you do the analysis, you have this quick view about gender quality, get some feel about the underlying data quality, make sure there's nothing weird. And then you can see this rate mapping, some mapping, rate mapping per tick, and so on and so forth. So again, each figure you can download to maintain by yourself. And then you have this duplicability. If everything looks good, essentially they output this mutation calling. So you see here we tell you the chromosome, the position, what's the reference allele, and then what's the filter, and so on and so forth. So you also have different format like MAFL. So this basically is a typical whole exome sequencing product where you probably will collaborate with your bioinformatician when you get the data. If they can retain results in say 1 week, I think you will be pretty happy. But now what I'm saying is with this kind of AI-driven tool, the moment once you get the data, you just kick out the project in seconds. Then you get the result on the same day. You see, here you get the VCFL on MFL format. You can save your own analysis, and then that could be directly go to downstream analysis, say identify mutated gene, do some mutation signature, and so on and so forth. So this is, say, not only this kind of AI-driven tool can do a higher level data processing, they could really run some fundamental basic pipeline to help us standard some RNA-seq, whole exome data, even single cell data analysis. So the other big thing we can think is about reproducibility. So for example, here is a paper published in MicroStratum in Nature, I think like 1.5 years ago. So this paper essentially presents some classical cancer genomic analysis. The paper actually sequenced more than 500 breast cancer whole genome sequence data. And then if you look at the paper, they generate some, here just some result directly from the paper. So you see, they provide you some basic statistics from mutation, indel, identify some driver mutation, and then they calculate some driver gene. They also deconvolute that mutation spectrum to infer the mutation signature. They even do some long non-coding RNA. So this paper is citation more than 1,000 times. So if somebody come to you say, hey, I'm interested in the paper using the method in my result, could you just first repeat this paper? Frankly, this kind of reproduce is a headache for bioinformatician. If I have a student that can repeat this, say, in 1 week, I will be very happy. I will consider that bioinformatician is very capable to repeat. But when you have this data loading in DrBioRight, where you have data load, you just ask the same question, could you do a mutation count? What's the driver mutation enriched and what's the mutation signature? You see on the right side, through this simple dialog, you essentially get the exactly same result from the published result. See mutation statistics, driver gene, mutation signature, non-coding. So that actually hints a new direction, how we essentially use this AI-driven tool. We can automatically repeat some analysis, even check the quality, especially for those computational-driven analysis part. Okay, so behind this, how you achieve this, I can show you this overall analytical flow. So essentially, we have this interface where people can either text or voice, and then we have this natural language processing module. They first given the text, they could identify what you want. So the survival analysis, disease name, the breast cancer, gene name, molecular data, gene expression. You tag those text, and then you're given this kind of information we feed to underlying model. We want to using predict what's the intention of the user. So that's the intention recognition. And then through the dialogue, usually we ask you to confirm this to make sure we really understand what you mean. Once you get your confirmation, understand your intention, DrBioRight will identify the data cohort and the analytical script to do the analysis. Once you call the analysis, they will check whether we have sufficient parameters to do the analysis. If all the parameters are fulfilled, then we submit this job. And then depending on this job, some job is very simple. We could probably do this in local browser. Some job maybe take a lot of computing resource, like those NGS or single cell data. We probably need to do fundamental mapping in cloud computing. But essentially, this job scheduler will do this job in given the most appropriate computing resource. Once you finish this job, they will collect those raw results, display that result through a module, visualization module. So given this visualization module, return this result into the dialog interface. From the dialog interface, we ask you to rate our job quality. It's good or bad, or you can report a bug. So you just say, I don't satisfy, this is my comment. We take this kind of user feedback to further update and train our model. So this is higher view, how we really implement this AI-driven tool. So in this case, from natural language processing, we develop a natural language understanding. So that's more like a brain to help you. It's just like your bioinformatics try to understand your question you raise. And then you have this kind of analytic toolbox where you have analytical module and then the visualization module, where we already implemented the most common bioinformatics tool in different language. Actually, you can also upload the U language. For example, no matter it's written in R, Python, MATLAB, because we use a ramper to really ramp that module. So the module really largely independent from the computing environment. And then we have this whole visualization module from mutation analysis, complex heat map, network view, pathway, and single cell analysis, and also pan-cancer analysis, and also this is box. So essentially, when you have the result, they could choose appropriate tool or module to help you discern the result. So in the future, what we want to do is really load more data. As I mentioned, currently we only have cancer genome data, but in future, a lot of standardized data, we can load more data. And also, we want to implement more pipeline, so people have choice to choose their preference. Second one, currently DrBioRight can only do the analysis, give the result, but we can further ask DrBioRight to interpret the result in the context of literature, because we can load the millions of the PubMed in that DrBioRight. So when he do some analysis, he sees some significant pattern, say breast cancer, in breast cancer TP53 expression associated with survival. They could tell you, hey, this pattern already been reported in 3 papers. Or they could tell you, this pattern actually contradicts in lung cancer. So given a result, the ability to interpret that in the current literature will greatly increase your confidence how to interpret the data. So the third thing is we want to get more social interaction. So currently we have this available. You can talk to DrBioRight. You can either by text or voice. In future, we are going to make not only one by one. You could make this group chatting. So you have one project, you invite your collaborator. You also invite DrBioRight as one member in the group. So when you do the analysis, you just add DrBioRight for second analysis. So all the people in that group can see the result and discuss together. We also have this more security. So when you log in, you really have public data, institution data, and the private data. So you don't worry. So given your security level, it depends. You can see how much data you can access. So if you have very high level data security, you can not only see public data, you also see private data. So you can lump them together, do your own analysis. But if you say only have do some public data module, some available public data, so when you log in, you can see the data only available to your security level. Finally, we want to make this platform transform to the smartphone. So that's we essentially remove all the time and the limit space. Essentially, nowadays everyone works remotely. I think with smartphone, now we can communicate this more, get a lot of flexible. So the major thing is currently I show you example, you may think, oh, those pipeline, I can do that. My bioinformatics can do that. But that's just the beginning. Currently, maybe you only have limit, but given the time, because you can constantly add a new job, a new pipeline, you can imagine in 2-3 years, and also because more and more people use the data feedback, we can more and more accurate interpret data, more accurate select algorithm. Essentially, this kind of AI journey will become very intelligent, very powerful, very knowledgeable. So that will be beyond any single person power. Okay. So after we published this paper early this year, actually, we got a lot of medium coverage, like Smart Brief. People talk about like how AI really changed data analytics, and then that got this positive view. But there are also some concerns, say this kind of concern, like are computational biologists losing their job and so on and so forth. So, well, first of all, I don't think our Dr. AI will replace computational job completely. But I do believe that's really certain AI essentially shifts the focus from running some routine analytic job, more and more new algorithm development, more tool development. So that's really the key thing. So I think we are in a very exciting time of innovation that none of them ever lived. So we, as a scientist, as a data scientist, we really need to transform ourselves, adapt to this new challenge, how to live in a post-AI world. We need to have this change our skill set and more importantly, mindset. So if you think this keep open-minded of this kind of thinking, this may potentially open new paradigm of research. So personally, when somebody doing research, it's more like he think idea, do some analysis, do some tests, write a paper, publish, okay? But nowadays, if you think you can start a question, immediately talk to this smartphone, do some immediate real-time analysis. And then this AI tool will help you assess the reproducibility, interpret this result in the literature context. And then because this is open source social media platform, you can easily engage different people, help you. Developer can provide some new algorithm for your test. Data scientists will point to some exciting new data. Biologists may provide a new interpretation. And the way you do this, you can, if we can couple this with a self-governing system, say some lab robot, you even can generate some new data and then feed this new data to the system to do new analysis. So you can imagine this new loop for research product. It combined with a lot of AI-driven power. So what I'm saying is, from now on, we may change our concept. Scientific research is more like a co-investigation between human and AI agent, rather than something people are using some AI-driven tool. So that's a fundamental message I was thinking when we move to that AI era, how we really use. So in summary, this part, I want to just quickly mention, we believe generation of data like DrBioRight really featured by natural language understanding. So language, only language help people to develop deep interaction, deep thought. So one cannot really have deep thought, deep interaction through a software graphic, right? But really through some conversation using natural language, we can push the deep thinking. So that's language understanding. Second one, we want to do some artificial intelligence. So behind that, we really have big data, no matter from data usage or query literature, you build a lot of model, choose the right data set, the right proper algorithm, good visualization module. And then everything you want to transparent. And then everything I believe future will not on the conventional desktop computer server is really mobile driven, because now that mobile is really the interface for your talk, then you outsource everything on big cloud computing on your institution server. So this essentially helps us make a intuitive, efficient, transparent, and collaborate. So this is essentially the first part of my talk. Any talk, question on to? Okay, so now I'll switch gear totally.
I have a question. Can I ask one? Hi, Han. Good to see you.
Hi. Yeah, good to see you. Yeah.
Yeah. I was just wondering, about the FDA just put up they have a regulatory framework for platforms that have AI, machine learning for patient systems, and it's worth looking at. It was just posted in January, and so because it's kind of software, they're thinking about it as software as a device, and this kind of thing. And so actually, if you hadn't, so I think by and large, I mean, you can make your system conform to it pretty well, but I don't know about the crowdsourcing. You know what I mean? That likely would be one that's not gonna, because there's got to be some kind of regular updates and versions having to do with the control about the training stats and this kind of thing. So in order to actually have this be useful as a clinical tool, there's likely going to have to be compliance with this, which might scale down the vision a little bit, and I'm just wondering if you thought about that.
Yeah, so that's a very good question, especially given our MD experience about Watson, So far, our BioRight not for clinical use, only focus on basic bioinformatics analysis, where you have very clear defined bioinformatics task, and then routinely that's handled by our analyst. And then we know the right answer. For this situation, because we know the clear right answer, we could easily benchmark the performance. And then because we generate a transparent report, everything can be trackable. So I totally agree with you. Go to the clinical side, like FDA, AI, it should be something should be take a great portion. But yeah, so that's crowd.
Yeah, so I guess the thing is just in terms of wanting to use it to kind of like start INDs and phase 1s and the early phase of the clinical, that would be something maybe something to talk about.
Yeah, that's really good advice.
Yeah. Thank you so much. Very interesting.
So that's really two parts. So this part really help people empower a lot of researchers can do things much faster if they have to waiting for their collaborator. So my second part is really about the pan-cancer analysis. It's more like a computational biology project. So I want to push an idea. So our ability to interpret it really goes through this pyramid. At the very bottom level, we need to get a better understanding about the genome structure. Once you get a better understanding, we can have better understand the genome biology. From there, we can have better interpretation about disease biology, how they really develop disease. Once you develop that understanding, you get a useful clinical application. So that's really the way. So that's highlight how fundamentally genome structure is important in our interpret big data. So in this case, we focus on a enhancer. As you know, enhancer is non-coding regulatory elements. They usually far from their target gene in the 1D space, but in the 3D space, that enhancer actually could bring to the promoter region, and then they could help recruit transcription factor and then turn on the target gene. So you can imagine that when you have inactive enhancer, this is well run around the nucleosome, but when you have active enhancer, this will polymerase II will bind to that region. Chromosome will open. Enhancer can transcribed by polymerase II. They generate some short RNA-seq read, say eRNA, enhancer RNA. We can call that eRNA. Okay. So we thinking that the eRNA signal could use as a readout for enhancer activity, because when you have enhancer activation, you have eRNA. But this kind of eRNA signal strongly depends on the annotation of eRNA locus. So about 2 years ago, we published a paper in Cell where we do the first pan-cancer analysis for TCGA, where you have near 9,000 patients. We use a phantom consortium. They using CAGE-seg, because CAGE-seg can really generate accurate precision. But that CAGE-seg only cover 15,000 enhancer locus. But even for that small size enhancer, we could quantify the enhancer expression level. And from there, we could develop an integrated approach to infer causal enhancer regulation. So the thing is, if we just get the enhancer gene, try to do a co-expression, that co-expression may or may not represent a causal effect. Say, but if you really tease out the causal model, we further consider the SNP in enhancer. So if you have enhancer really cause the gene expression, you would expect to observe the EQTL signal for the SNP on the enhancer. But on the other side, if a co-expression due to the gene-regulated enhancer, that's a reactive model, you will not see that. On enhancer and the gene co-regulated by third factor, you will not see this EQTL signal. So essentially what we did is we have 33 cancer types. We first identified those highly co-expression enhancer and the gene pair. And then we further incorporate the EQTL signal from GTEx and 1,000 genome. You sort of can really see there's one SNP in enhancer, they really cause gene expression level. So that's called enhancer gene regulation. And then we further have this high-seq 3D genome to make sure that proposed enhancer physically close to target gene. So that's really direct causal enhancer regulation rather than some secondary effect. So with this approach, we could build a gene enhancer regulatory network. So in this network, especially focus on the therapeutic target. Each circle represents one therapeutic target. Each box represents one enhancer. And then one pair we inferred got a lot of attention is this PD-L1. As you know, PD-L1 is a major therapeutic target for immunotherapy. And then we found that there's a chromosome 9, there's a small short enhancer. We can call it enhancer 9. Which enhancer is a direct regulated PD-L1. So we can further validate this, say, given cell line, we can use a CRISPR to knock out this region in enhancer 9. And indeed, in both this messenger level and protein level, you see when you delete that enhancer 9, even that only very short, you substantially reduce the PD-L1 expression at both messenger level and protein level. Even with the intergamma stimulus, you see the PD-L1 expression level still become much lower. So that's if you, and then through some further data mining, we basically bring a model, NF-kappa B mediate promoter interaction. Basically, NF-kappa form a dimer, bring this enhancer 9 close to the PD-L1 promoter, basically turn on the PD-L1. So that's just give you one example. When you have this detailed understanding about how important therapeutic drug target be regulated, they could potentially develop new target. But the problem is, for this study, we only focus on very small set, well-annotated enhancer locus. So that's due to the phantom. But for large mass majority region, for example, for super enhancer region, you know, when Richard Young really bring this concept, people know super enhancer region is really big chunk region in the human genome, but they really provide very important role in control cell identity, and tissue development. So really, when you superimpose those fetal modification signal, you see some region have very dense chip signal. So their region can define as a super enhancer. So through study, you have quite a bit super enhancer region. The problem is, when you look at the super enhancer region here, just give you this genome region, represent genome region, you have this lot of fetal modification peak, and then you define this region. But if you look at the scale, this region easily several kb. For such a big region, essentially, you're very hard to quantify eRNA signal. Because when you have this big signal, remember ER signal is very weak. So when you have big region, you just have a lot of background noise. That background noise will easily overcome those true signal. So essentially you get nothing. So a fundamental idea to understand the super enhancer activity is how can we accurately quantify eRNA signal? Let me repeat, the challenge is how to quantify super enhancer activity is how can we accurately quantify ER signal transcribed from super enhancer region. So from this, we recently published a paper presented this map. What we really did is we studied from a huge amount of data aggregation, try to understand what's the principle governing super enhancer region. From there, we do some global, identify some transcription unit in the super enhancer region. Then we do some downstream application. So what we did is when we get a set of core super enhancer region, meaning the super enhancer active across multiple tissue. So that represents the most confident super enhancer region, only 5 megabit region. And then we can easily aggregate hundreds of TCGA samples together. And then we found this region, when you aggregate 100, you see some very clear peak. So here you can see this colored by different peak. And then they actually form this kind of unit. So this is, when you have this peak, they form this very clear expression peak in different cancers. Sometimes there are multiple peaks. So you have this quite peak. And then we also want to see this peak location is conserved across different data sets. So you can, using the same practice for GTAX, where you have normal tissue. You found this for this region, the peak location actually quite consistent. Even you use a totally different consortium product, totally different data, you see the peak location actually quite consistent, meaning that's really not from some random noise. And then further, when you superimpose this kind of peak, what we really found is they form this very sharp, really just about 100 base pair peak. So this happens in both TCGA on the left side and then the GTAX on the right side. Across different disease cancer type, across different tissue, you see eRNA form this very sharp peak. And then we also wonder, because this only 100 base pair, this reminds us the ribosome, nucleosome, the nucleosome ramping is only 100. So we're also looking for this peak location. We found for those peaks, they are flanked by tRNA transcription binding motif. So on the both sides, you see a very clear motif on that peak side. So that also highlights this really potential due to an enhancer where they enrich some important motif. And then we further collect those because we wonder what regulates this peak. One hypothesis is this regulates really due to the underlying nucleosome. So we collect the nucleosome profile generated by NMNase. So from 29 different tissues. And then when you have this nucleosome position on those peak, you see there's a well-positioned peak corresponding to that peak. So I want to make it clear because we do multiple tissue. So when there's a nucleosome occupancy, there's no that peak. But when this nucleosome off, you see there's an activation for that ER signal. So, but when you superimpose the signal across multiple tissue and then the nucleosome position, you see a well-positioned nucleosome corresponding to that peak. So that's really, if you look at that peak position across different lineage, because in the mouse and the peak, they also generate some nucleosome binding profile. You found even the sequence actually not conserved. The nucleosome position is quite conserved. So now we can see those eRNA peak we observed actually due to some very evolutionary conserved, well-positioned super nucleosome. So based on that, we can propose a model how that eRNA really, you see that eRNA peak is really when you have this well-positioned nucleosome here highlighted in red, in green color. So when this nucleosome occupant that you turn off that eRNA signal. But when you have this activation, the polymerase II recruit some transcription factor binding to that. They have some transcription activity. You observe some eRNA signal. But because that eRNA signal is such consistent from that locus, you see a peak when you observe across hundreds of samples. But when you look at individual, you don't have this power because that very weak transcription easily get lost in the background. So that's really the working model for our understanding the eRNA signal. Now the hypothesis, how can really have two observations. One is this kind of super enhancer region contain multiple eRNA locus where they can generate a very sharp eRNA species, only 100 base pair long. Second one, when you have this eRNA locus, they potentially coincident with well-positioned nucleosome. So that's two important observations for such kind of super enhancer locus. So we then want to do the global discussion. We extend our analysis to a 370 megabase super enhancer region. And then we studied some nucleosome position where basically you look at the PCA. There's a one PCA component called PCA3 that corresponding to the nucleosome position. So to make the story simple, basically a negative PCA3, meaning that position have well-positioned nucleosome position occupancy. So based on that principle, based on the local expression peak, based on that nucleosome, we actually identify 300,000 eRNA locus in that super enhancer region. So this is each locus have very clear star endpoint, only 100 base pair long. So now you have extremely high resolution eRNA locus. Then we can validate this, say, given those locus, for certain region they have eRNA signal, you see they have very clear CAGE signal around the eRNA peak we annotated. But for the mass majority region, because you don't have the CAGE signal, for the mass majority, our eRNA signal really can give a lot of new locus. So the reason is why.
Yeah, sorry to interrupt. Like, I just want to give you, we have till 5. So
yeah, just 2 minutes. I will run out.
Yeah.
So, yeah.
Sorry about that. Yep.
Yeah. So basically, we have this. Why we believe enhancer could be, because we believe enhancer is a good way to address the tumor heterogeneity. You can imagine that what we got is a bulk sample. If you want to do gene level correlation, say, essentially you do gene expression with phenotype, what you look at is aggregation different cell type. So when you do this phenotype correlation, this correlation is very weak. But suppose you do enhancer, each cell type have their specific enhancer. Enhancer A control do gene expression. So when you do the enhancer, suppose this phenotype is controlled by enhancer A. When you do enhancer level, you would see a strong correlation. In other words, if you do gene level, because we usually use a bulk sample, you essentially confounded signal by different cell type. But when you do the cell type enhancer, you really have this power. So for example, if you look at we want to predict, say, immunotherapy. So you have the melanoma patient, same PD-L1 treatment, some get respond, some not get respond. So if you look at the paper, if you look using the coding gene, on the left side, you see there's no significant gene because the sample size is limited. Really, you see a uniform p-value distribution. There's no significant gene show. But if you look at the enhancer, you really see a significant p-value enriched. Even given FQ f 0.05, you have about 164 locus. And then if you do this heat map, this eRNA signal can really separate these two clinical response. And then if you do a gene site enrichment based on the enhancer, you can really see that the CDX, this enhancer regulate the biology is CDX T cell exhausted on CDX depletion. So this perfectly match what we understand how T cell understand play role in the immunotherapy. So this just give you an example because we use specific enhancer that probably can inform T cell, so we can make this clear phenotype inference. But when you do the gene level, because you essentially get aggregate signal from across all the cell type, you lose the power. So this kind of pattern enhancer also can be validated by independent cohort. So what I'm saying here, basically say, super enhancer get discrete enhancer signal locus by eRNA peak. And then this eRNA peak expression regulated by dynamic well-positioned signal. Our analysis provide a high resolution map for this eRNA locus. So people can easily use RNA-seq to quantify super enhancer signal. And then we believe eRNA signal provide a much better inherent power for quantitative traits beyond gene expression. So, and then I think we're close to time. I just want to sum. The first part is really my talent fellow Jun Li lead this effort to make this very powerful analytical tool. My second part is really my postdoc fellow Han Chen make this enhancer analysis help provide. And then I will thank my funding resource and stop here. Here is my favorite sentence share with you. The best way to predict future is to create. So yeah, so I will stop here. Happy to answer any question.
Thanks a lot, Han.
Sure, I got a question.
Sure, go ahead, Brian.
Yeah, hi, Han, thank you so much for the second part of the talk. It was fabulous. You know, one of the things that has been confusing me a bit is the super enhancer really involves a number of enhancers, really kind of 3 to 40 genes that are likely on, you know, adjacent pads or at least, you know, spatially close. So, you know, that's one thing that I think is an opportunity for some continued analysis, because focusing on a single nucleosome transition, which is terrific, gives you one enhancer, but doesn't give you a super enhancer, doesn't describe how that gets in and out of the transcriptional condensate. Not a criticism. It's just next steps.
I totally agree. I think that's once we fundamentally understand this map, the next step is really how to see their synergetic effect, aggregation effect, and then build a model. So that's actually perfect, exactly what we try to see, how a gene, there upstream have multiple enhancer units, how they signal really talk to each other, determine a specific cell type expression profile. That's totally on our agenda.
Yeah, so that would be good to talk. I would love to have a chance, and Vera and I are collaborating, love to have a chance to be able to speak with you more about that.
Sure, that would be great. Yeah.
Thank you so much.
Yeah.
So Han, I had a question about the first part, the DrBioRight.
Yeah.
So is that sitting on what databases is it querying for doing analysis at this point? You plan to, so 2 parts. One is
so currently we have some widely used the data like TCGA, SCGC, CCLE, each of the 100. We are planning to add more data set, like we have this pediatrics, and then you have the gene. So we can essentially add more data set to that. Just because my group has limited manpower at this moment to try to make the basic function infrastructure more stable. Yeah, but definitely we can add more data. And also, actually, one way is if your current data not available, we are going to record this. If there's sufficient user querying a data set which is not available, we are going to add those data to the platform. Yeah.
Are you participating in CP-TAC?
Yeah, that's actually one thing we're going to do, that to try to add the CP-TAC to the data set. Yeah, because I can see a lot of people want to do that joint analysis between TCG and CP-TAC.
Can you conduct your analyses from the existing proteogenomic data we already have?
Yeah, that's actually, once we load the data, all those module, visualization module will already available. You can easily say, if you ask a question, we can now ask a question, what's the correlation between 2G at a messenger level? We can easily, by load the CP-TAC data, we can ask what's the correlation between 2G at the protein level? What's the correlation between messenger and protein level? So all the questions should be easy. Yeah.
Yeah, I'm Gil Oman. I'm part of the CP-TAC.
Yeah, Gil, I know. Yeah, actually,
your name.
You are the leader for, if you can. So because I'm involved in
yeah, no, I'd love to see you capture you into this. Yes.
Yeah, you can talk to them if you can CP-tag consortium load a lot of data, so people can easily just query it. So basically, now they have any question, they just come to DrBioRight to say, what's the TP53 correlation mutation with, say, P10 protein in CP-tag? They will get result immediately.
Good, good.
Yeah.
Han, are you exploring some of the biological implications for the eRNA storm? I mean, you know, there's probably other things that it does.
Yeah, that's actually, so I think there's a eRNA control the gene and then confer the specificity. And then we are now doing a lot of like the clinical application, try to see eRNA, whether they could, as a potential biomarker. Fundamentally, for those therapeutic target gene, we try to see how the gene pattern is regulated by enhancer. Because when people really think how the oncogene boosts up on tumor suppressor, they usually think it could be due to copy number, they due to mutation, they due to demethylation, they due to gene fusion. We believe there's one more way is try to disruption of the enhancer regulatory elements, also another powerful way to perturb those key genes. Yeah, that's one.
Thank you.
Yeah.
You know, just one more. So when you show the scans on the nucleosome positioning, you know, and you show all those kind of like scans from the different tissue types, you know, it's hard to kind of tease them out. But then when you showed the other slide, you know, that showed the nucleosome positioning profiles, at least on the center nucleosome, as a function of different species and things like that. You know, it did show a lot of you know, a lot of variability actually. You know, there was so is there something that we could learn from all of that? Because I was
yeah
thinking about that.
So there are several things. Number one thing is when we have this profile, the nucleosome profile, there's no such systematic cohort. We are collecting those nucleosome data from different literature. So it's a meta-analysis. You can imagine, even though we make a lot of effort trying to remapping, because they are not like a consent, they could have some variation noise. So for that part, we really want to see some general pattern. Yeah, I agree, there could be noise. One thing, because there's some fundamental noise when we do this kind of aggregated cohort. The other is when we infer that enhancer for sample size, maybe there's some variation when we define the boundary of that, because intrinsically eRNA expression level so low. So even we have hundreds of them, we can see shape, the boundary maybe not vary. So that's another thing, yeah. So when you, yeah.
It's fantastic.
Right, great.
Really great talk.
Yeah, thank you. Yeah, I'm looking for, I have several colleagues scheduled. We are talk tomorrow. We can have one by one discussion. Yeah, hopefully you enjoy. And then the best way just use, talk to Dr. Wright. I show you that. Yeah.
Yeah, I did try it on the side, by the way. It was very cool. So on that note, if there are no more questions, thanks a lot, Han, for taking.
Thank you, Vera. Talk to you later. Bye bye. Yeah, Wally. Yeah, all right. Bye bye. Yeah.